Imagine trying to build like a microscopic factory inside a room that is already just packed wall to wall with someone else's machinery.
Right. And you can't build any walls to claim your own space.
Exactly. You don't have any tiny trucks or conveyor belts to move your parts around, and yet you have to somehow establish an assembly line, manufacture delicate parts, pack them with, well, explosive amounts of genetic material, and get them out the door.
Which is an absolute engineering nightmare.
It really is. And that is the exact nightmare a bacteriophage faces when it hijacks a cell. Welcome to today's deep dive, where we are shrinking down to the microscopic level to witness this hostile takeover firsthand.
And we're specifically looking at how a bacteriophage virus called SPI1 infects a really common soil bacterium called Bacillus subtilis.
Yes, and our source material today is this fascinating paper from Nature Communications. They use some truly cutting-edge imaging to basically freeze these cells in time and look inside them in 3D.
Yeah, the technique is called electron cryotomography, or cryo-EO. And the way they capture this factory in motion is just as important as what they found.
Right, because normally if you want to look at the inside of a bacterial cell, the cytoplasm, you'd use chemical fixatives, right?
You would, but fixatives warp the delicate structures. The cytoplasm isn't just like a swimming pool of water. It is a dense, highly viscous gel packed with proteins. So to see it in its native state, they literally plunge freeze the infected bacteria in liquid ethane.
Liquid ethane, that sounds intense.
Extremely. They do it so rapidly, we're talking a fraction of a millisecond, that the water molecules inside the cell simply don't have time to arrange themselves into ice crystals.
Because ice crystals would act like microscopic daggers, just shredding the internal architecture you're trying to study.
Exactly. It forms this glass-like solid, freezing the biological action perfectly. Then they use a focused ion beam, which is essentially a molecular sandblaster, to mill away the sides of the bacterium,
leaving just an impossibly thin slice of the cell to look at under an electron beam. So we aren't just looking at a pulverized cell soup, we are walking the actual factory floor.
We are. And the first thing the virus has to do on that floor is clear some space.
Okay, let's unpack this. Because normally, Bacillus subtilis is just crammed with ribosomes, right? The host's protein-making machines.
Right, they're everywhere. But around 15 to 25 minutes after the virus injects its DNA, the interior architecture drastically shifts. The virus creates this massive dedicated zone right in the middle of the cell,
which they call the vDNA compartment, right? Viral DNA compartment. It kicks out almost all of those bulky host ribosomes to make room for its own blueprint.
It does. It reserves this huge area entirely for replicating its own viral DNA.
But wait, usually when we think of a compartment in biology, we think of a wall, like the lipid membrane around our own human nuclei, or a protein shell. Does this virus actually build a wall?
What's fascinating here is that there is absolutely no physical wall. There's no membrane, no protein cage.
Wait, really? How do you keep thousands of giant protein factories out of a space without a barrier?
It relies entirely on biophysics. As the viral DNA replicates, it forms what are called concatemers.
Concatemers. Let's define that for everyone listening.
Sure. So imagine a massive continuous molecule made up of multiple copies of the viral DNA sequence, all just linked end to end.
So instead of neat little individual rings, it's one impossibly long tangled web.
Exactly. And this web is so dense and highly compacted that it physically excludes large complexes like ribosomes. The ribosomes are just too big to diffuse into that tight mesh.
Oh, wow. So it uses the sheer density of its own genetic blueprint to just shove everything else out of the way.
That and a phenomenon called liquid phase separation. The viral DNA basically condenses into a distinct liquid phase within that crowded gel.
Oh, I love this concept. It's like pouring oil into water, right? Or shaking up a vinaigrette. The oil eventually pools together into distinct droplets that separate from the vinegar without needing a plastic bag to hold it.
That's a perfect analogy, except the oil here is a highly charged, entangled web of genetic material. It creates a physical boundary defined entirely by molecular crowding.
Yeah.
It's basically a phantom room.
A phantom room. That is incredible. Okay, let's do a quick recap before we move to the next stage of the factory.
Good idea.
So, bullet 1. The SPP1 virus hijacks the bacterium and needs workspace. Bullet 2. It creates a massive vDNA compartment in the center for its genome. Bullet 3. This compartment has no walls, but uses phase separation and sheer density to kick out host ribosomes.
Spot on.
So here is a question for you, the listener, to chew on. If you had to clear out a crowded room without building a physical wall, how would you do it? Because the virus does this simply by taking up space with an impossibly dense web.
It's very efficient,
Yeah,
but it does create a logistical problem.
right. Because if this wall-less vault in the center is just for DNA, where are the actual virus bodies, the empty shells or capsids being built?
You wouldn't want to build delicate, empty geometric shells right in the middle of a chaotic, tangled web of DNA.
No, you'd want a clear workbench. And the cryo-ET images showed they aren't built in the center at all. They're built directly attached to the inner side of the bacterial cell membrane.
Right. The virus uses the host's inner wall as its scaffolding. And to see how this works, the researchers looked at 3 specific viral proteins.
Let's give them an architectural mnemonic to make it easy to picture. We have GP6, GP11, and GP13.
To track them, the researchers genetically tagged the GP6 protein, which is the portal protein, with a fluorescent marker. Then they used a specialized microscopy technique that only illuminates a tiny sliver of the cell right at the membrane.
Oh, so they were essentially just looking at the wallpaper of the room, ignoring all the glowing stuff in the center.
Exactly. And they saw that early on, these GP6 portals were just zipping around. But when assembly starts, they hit the brakes and lock into place directly on the membrane.
So GP6 is the anchor. If this is a house, GP6 is the single door frame bolted right to the cell membrane. And once it's bolted down, it calls in the next player, GP101.
Right. The GP11 proteins are the internal scaffolding. They gather around that bolted door frame and build a temporary curved internal support structure.
And that scaffold guides the third player, GP13, which are the actual bricks, the outer tiles of the shell.
Yes. The GP13 bricks lay down over the scaffold, growing outward from that single anchored door. It grows in this precise curve until it finally closes back in on itself,
forming a complete empty sphere, about 55 nanometers wide, which they call procapsid I.
And here's where it gets really interesting. The moment that sphere fully closes, a structural shift happens, and the whole thing pops right off the membrane.
It just detaches and floats away. But why anchor it to the wall in the first place? To figure that out, the researchers created a mutant virus missing that GP6 portal protein, the SPP1-GP6 mutant.
And the mutant was a total disaster. Without the GP6 door to anchor the process to the membrane, they found incomplete shells just floating aimlessly in the cytoplasm,
forming random, incorrect shapes, spirals, tiny useless spheres.
Completely useless. The membrane is acting as nature's ultimate quality assurance manager. By starting at the wall, it guarantees only 1 portal door is installed per shell.
Because if it tumbled around in the open soup, 2 doors might try to build on opposite sides, and the whole geometry would fail.
And since that single door is the only way DNA gets inside later, anchoring it is just a brilliant structural control strategy.
Okay, time for our second mini recap. Let's lock this assembly line into memory.
Let's hear it.
Bullet 1. Virus assembly starts at the inner cell membrane, not floating randomly. Bullet 2. The portal protein, GP6, is the mandatory anchor point. Bullet 3. Scaffolding GP11 and shell bricks GP13 build around the portal until the shell closes and pops off.
Perfect.
So for you listening, think about this. Why do you think the virus chooses to build its shell attached to the wall rather than in the middle of the room? Maybe it's just to keep the delicate open shell away from that chaotic center web until it's structurally sound.
It makes a lot of sense. But now we have an empty, closed chassis floating around,
right,
and the DNA is still sitting over in the center vault.
Right. How does this empty balloon, this procapsid I, get to the phantom room?
It migrates there. But what's wild is that they proved, using more mutant strains, that this happens even if the virus's DNA packaging motor is totally broken.
So it doesn't swim there. There's no tiny propeller.
No motor at all.
Yeah.
The outer surface of the empty shell just has a natural biochemical affinity for that dense DNA phase. It naturally diffuses toward it and just sinks right into the vDNA compartment.
It essentially just parallel parks itself into the vault using physics.
Okay.
Okay, so now we are in the vault. We have to drop the engine in. We have to get meters of tangled concatenamers stuffed into a tiny 55 nanometer sphere.
This is where the sheer biophysical violence comes in. The virus uses a terminase motor made of proteins GP1 and GP2.
And I'm guessing this motor docks right at that single portal door we so carefully built.
Exactly. It docks at the portal, grabs a strand of the viral DNA, and starts furiously pumping it inside, base pair by base pair.
But wait, DNA is highly negatively charged, right? So if you shove meters of it into a microscopic ball, those charges are going to violently repel each other.
The pressure is immense. We are talking tens of atmospheres of internal pressure building up inside that tiny shell.
Which triggers my absolute favorite part of this paper, the capsid expansion.
Yes. The shell doesn't just passively take this pressure. The entire protein lattice physically shifts. The 55-nanometer sphere violently snaps outward.
Expanding into this larger 65-nanometer shape, they call it procapsid 10.
And the geometry changes entirely. It goes from a smooth sphere to a highly angular, thin-walled icosahedron.
Like a 20-sided die from Dungeons & Dragons. If you want to picture this, imagine a slightly deflated, crumpled geometric camping tent.
Right.
And the moment someone starts forcefully stuffing an incredibly thick, stiff rope inside, the pressure builds until snap. The tent pops violently outward, stretching into its final, rigid tension shape.
That's exactly it. And when it snaps outward, the physical expansion literally ejects the GP11 scaffolding proteins that were still inside.
They just get kicked right out through the pores of the shell.
Yeah, which brilliantly frees up even more internal volume for the rest of the DNA. The motor just keeps pumping until it's a completely loaded, rigid icosahedron.
It is a microscopic miracle. It doesn't just burst. Okay, recap number 3.
Let's do it.
Bullet 1. The empty procapsid eye sinks into the vDNA compartment using chemical affinity. Bullet 2. The terminase motor starts aggressively shoving DNA inside. Bullet 3. The intense pressure causes the shell to snap open, expanding into a larger, angular icosahedron.
Beautifully summarized.
So, question for the audience. Imagine the incredible pressure inside that tiny biological shell. How does a lattice made of just proteins hold back tens of atmospheres of force without shattering? The structural engineering is just mind-blowing.
It really is.
Yeah.
But having a rigid shell full of pressurized DNA is only part of the job. A bacteriophage needs a tail to infect the next cell.
Ah, right. The iconic needle-like tail. Are those being built in the DNA vault too?
No, and if we think about cellular logistics, trying to build long, flexible tails inside a tangled web of DNA would be a nightmare. They'd get hopelessly entangled.
So the virus uses spatial segregation again. It moves to a new zone.
Precisely. Once the capsid is totally full of DNA, it mysteriously changes its affinity. It no longer wants to be in the dense vDNA compartment. It physically leaves the center and migrates out to the periphery of the cell, right under the membrane.
So it goes from the loading vault in the center out to the shipping docks at the edge.
Yes. And out at the periphery, the virus has been running a separate assembly line, building these delicate tails.
So the full head meets the pre-assembled tail at the edge. They dock together at the portal, and boom, you have a mature infectious virus.
But there is one massive risk here.
What's that?
If you have hundreds of these highly sticky, mature viruses piling up at the shipping dock, wouldn't they just jam together?
Oh, like if you covered a bunch of tennis balls in Velcro and threw them in a box, you'd just get a useless, solid clump.
Exactly. And the researchers proved this by looking at one final protein called GP12, which coats the outside of the mature capsid.
Let me guess, they mutated it.
They did. They created a mutant lacking GP12, and the resulting viruses stuck together incredibly tightly. They formed these massive, highly ordered 3D paracrystalline grids.
Like perfectly arranged cannonballs.
Right, which they called warehouses. And biologically, a warehouse is a dead end. If they're locked in a solid crystal, when the bacterial cell finally bursts, they can't disperse. They'll just sink as a heavy clump.
So GP12 acts as a vital spacer. It physically prevents the viruses from getting too close and locking together.
It keeps them loose and ready to deploy.
Let's do our final mini recap before we look at the big picture. Bullet 1. DNA-filled capsids exit the center zone and go to the periphery. Bullet 2. They attach to pre-assembled tails at the edge. Bullet 3. The GP12 protein acts as a spacer to stop them from jamming into a useless crystal.
Exactly right.
So, audience, you now see the full map of this infected cell. The membrane, the center, the edge. How much could our own human supply chains and factory designs learn from this kind of spatial efficiency?
It is a perfectly optimized production line segmented into highly specialized zones.
So what does this all mean for us? Why spend millions of dollars slicing up bacteria with ion beams? Well, understanding this map reveals vulnerabilities.
Right. If you know how the factory floor operates, you know where to throw a wrench.
Exactly. If we know an empty shell physically must migrate from the membrane to the center, and a full one must go from the center to the edge. If we can develop a drug that just changes the stickiness of the virus, preventing it from moving, the whole infection breaks down.
We wouldn't even need to destroy the virus. We just stop the conveyor belt.
Exactly. We just jam the logistics.
But this actually raises an important question, maybe the most profound mystery in the whole paper.
Oh.
You mentioned a conveyor belt. But There is absolutely no evidence of a dedicated transport system moving these parts around.
Wait, really? Because, like, in human cells, we have a cytoskeleton. We have literal motor proteins that walk along little molecular highways to carry cargo.
Right, and some jumbophages even build tracks inside bacteria. But SPP1 doesn't have any of that. No molecular trucks, no little walking proteins.
Then how is it moving so flawlessly between these specific zones? From the wall to the center to the edge?
The hypothesis is that they move purely by altering their surface thermodynamics.
Oh, wow.
When the shell is empty, its atomic arrangement naturally diffuses into the dense DNA web to find its lowest energy state. But when the motor pumps the DNA in and it violently expands into that 65-nanometer icosahedron.
That subtle shift in shape and charge changes how it interacts with the fluid.
Exactly. That physical change alone naturally expels it from the DNA phase and pushes it out to the periphery.
That is chillingly elegant. Without any tiny trucks, without any central brain directing traffic, these viral parts just know where to go simply by expanding and changing their surface charge at the exact right millisecond.
It's all driven by local crowding, phase separation, and the blind laws of physics.
It's incredible. It really makes you reconsider the boundary between biology and chemistry.
It's very humbling.
So I want to leave you, the listener, with this thought. If biophysics alone, just shape, charge, and pressure, can perfectly sort and assemble an alien factory inside a crowded cell, what else is happening inside our own bodies right now? What complexes are sorting our molecules and building our structures entirely without a central brain to direct it? There is this hidden thermodynamic choreography to life that we are only just beginning to capture on camera.
It's a whole new way of seeing biology.
It really is. Thank you for joining us on this deep dive. Keep questioning the microscopic world, and we'll catch you on the next one.